Hello and welcome everyone. In this podcast, we're going to focus on energy that is produced in the mitochondria. And more specifically, we're going to talk about oxidative phosphorylation. This is called oxidative phosphorylation because it requires oxygen and a phosphate is added to ADP to create ATP. Before we talk specifically about oxidative phosphorylation, let's think about where we've been. Remember, a few podcasts ago, we started off with glucose and talked about how we could get glucose into the cell, and then how, through glycolysis, we made pyruvate. We said in this process, if you remember, we made some ATP and we made some NADH. And we mentioned how pyruvate then could be turned into or converted into acetyl coenzyme A. In this process, we talked about how we made some NADH. We then talked more recently about how acetyl-CoA could be fed into the citric acid cycle. In the citric acid cycle, we made NADH and FADH2. We also made 1-GTP, remember, and then that was converted into ATP. So now we're at the end here. What are we going to do next? The glucose that originally entered the cell has been completely oxidized through the citric acid cycle. Yet, there is still a lot of energy left that has been stored in NADH and FADH2 made there, here, and in glycolysis. What we're going to talk about next is how we harness this energy from these activated carriers. And the process that's going to use these activated carriers is called oxidative phosphorylation. I'm going to talk a little bit about oxidative phosphorylation just to introduce it to you, and then we're going to expand on that throughout the rest of the podcast. First of all, it occurs in mitochondria and in aerobic bacteria. During oxidative phosphorylation, ATP is produced, and it does so by transferring electrons from initially food, but ultimately what we're going to work with in oxidative phosphorylation are these activated carriers. So we're going to get it from NADH and FADH2. And we're going to transfer those electrons ultimately into oxygen to form water. And what makes this process different than the chemical reactions we talked about in glycolysis and the citric acid cycle is that we're going to transfer these electrons through a membrane. So this is membrane dependent. Glycolysis occurred in the cytoplasm. Citric acid cycle occurred in the matrix, independent of the membrane. Oxidative phosphorylation requires a membrane, and we'll talk about that as we move forward. But before we move forward with oxidative phosphorylation, I think it's a good idea to talk about where those membranes occur. And I'm going to focus on eukaryotes. And oxidative phosphorylation occurs over the membranes of the mitochondria. In bacteria, it occurs at the inner membrane. But again, we're going to focus on the mitochondria here with eukaryotes. So if we were to look at a typical eukaryotic cell, what we would see are these mitochondria that are these very long tubules, interconnected, highly branched membrane structures. And we know that these mitochondria at one time were independent bacterium. And they, just like the eukaryotic cells, they had their DNA inside of them. Just like we have DNA inside of these mitochondria. So what we believe happened was that at some point early on in the evolution of eukaryotic cells, a primitive anaerobic eukaryotic cell engulfed this bacterium. And over time, this bacterium became this structure known as the mitochondrion inside of our cells. If this event didn't occur over 1 billion years ago, it's hard to imagine that we would have ever evolved into a multicellular organism. It would have been hard to imagine us having a different mechanism to obtain all that energy that's necessary for survival as we know it. What I want to do is to examine a cross-section of the bacterium. So we're going to take that tube and just cut a little piece out and look at it on its side. And so this is what you're probably used to seeing in various textbooks and classes, is this classic kind of structure of a mitochondrion, where you have your outer membrane, your inner membrane, this space right here in between the two membranes, we call that the inner membrane space, and this part in the middle we call the matrix. Within the matrix we have mitochondrial DNA that is attached to the inner membrane, but it's in the matrix. Throughout the rest of this podcast, we're going to talk about how oxidative phosphorylation occurs across this membrane. And throughout oxidative phosphorylation, we'll learn that we make a lot of ATP. Since it is the mitochondria that generates most of the ATP in a cell, sometimes the mitochondria are localized to very specific regions in a cell. For instance, in a sperm cell, where you have the head of the sperm here and the flagellum here, there is this base here, contains a lot of cytoskeletal structures that we call the axoneme. Around the axoneme, we see mitochondria wrapped around it, providing the necessary energy for the motility of the sperm. It's specifically localized here because that's where the energy is necessary to move the flagellum and ultimately the sperm. We don't see mitochondria down here, and we don't see mitochondria in the head of the sperm either. It's only in this very specific place where the energy is needed. We also see it a lot in muscle fibers. So within the fiber itself, there are these muscle fibrils that are responsible for muscle contraction. And we will see large amounts of mitochondria associated right here in between and wrapped around these fibers, providing that necessary energy for muscle contraction. We also see them at the neurosynaptic cleft junction in neurons and other places, it's very specific locations. But the key is you expect to see mitochondria not in only cells that produce a lot of energy, but in specific parts of those cells that produce a lot of energy. Now, in some cells, there are more mitochondria than in other cells. So, for instance, an oocyte contains a very high concentration of mitochondria. And I'm going to draw these mitochondria in the oocyte a little differently. Not as large tubules, but as almost punctate spots. They have a little tubular function to them or resemblance to them, but if you looked at an oocyte, it would almost look like it's just full of little tiny mitochondria, like so. Now you might say, well, Gorsuch, you just told me they were tubular shaped. Well, that's true, but during some specializations of cells, the mitochondria will take on a different structure, such as the oocyte. Why they form these structures, we don't have time to get into. I can always share that with you later, but I think we want to move on. Because the main point I want to make here is about energy. And that is, if you take any particular cross-section through an oocyte, you're going to see tons and tons of mitochondria. Because that mitochondria is necessary for those very early stages of embryonic development. In fact, we know if an oocyte has fewer mitochondria in it, the chances of it surviving after fertilization are reduced. We know in cancer cells are also chock full of mitochondria. They're not typically going to be spherical like we see there in the oocyte. These will be more classically shaped. But they're going to be all over it. Again, the theory is that the mitochondria provide that energy necessary for that uncontrolled growth that is characterized by cancer. Now, in addition to the relative amounts, now some cells will change the amount of mitochondria they have in them. For instance, muscle cells. Over a course of the lifetime of a muscle cell, there may be more or fewer mitochondria in them because the muscle demands may change throughout the course of a lifetime. Okay, now what I want to do is I want to go back to this membrane here this membrane here, in the inner membrane, and talk about how we're going to generate energy using this membrane. Okay, so let's spend some time talking about oxidative phosphorylation. We can think of oxidative phosphorylation as occurring in two stages. This first stage of oxidative phosphorylation is characterized as moving protons across the membrane, across the inner membrane, either the inner membrane of the mitochondrion or the inner membrane of bacterium. It moves these protons across the inner membrane, these hydrogen ions across the membrane, it uses the energy from electron transport. So if we draw a membrane here, and we want to get these protons to this other side, we have a few problems. Some of the problems are that these kinds of charged elements, atoms, do not readily go across the membrane. We also have a concentration problem. There are more protons on this side of the membrane. It has the more positively charged structures on this side. So this energy demanding step requires some energy. And we'll talk about this in almost exhausting detail, but it's going to use electrons as that energy source. So for now, I'm just going to draw this membrane protein like so, and draw this proton going across the membrane. In doing so, it's going to use these high energy electrons to generate the energy necessary to move that proton across the membrane. By pumping these positively charged protons over here, we've established a very strong electrochemical gradient. And this electrochemical gradient has great potential. And we're going to see what that great potential does in this next stage. So this next stage of oxidative phosphorylation, we move the protons down the gradient through the membrane using ATP synthase. So we'll draw that here. And so these protons that are currently on the higher concentration on this side will move through this ATP synthase, and that energy that's created from their movement across this enzyme will convert ADP plus PI, which are in the active sites of this ATP synthase. These protons generate the energy to convert this unfavorable reaction, ADP plus PI, into ATP. And then ATP is released and leaves the mitochondria to do cellular work. Now I want to go back through each of these stages again and spend a little bit more time talking about some specifics of how energy or electrons are moved and generates this electrochemical gradient. So let's talk about our friend here, NADH. I'm going to draw the structure here, but I'm going to draw it for the purpose of showing you where those high energy electrons are. Those high energy electrons are going to be associated right here. Remember, before this is NAD+, and so the hydrogen is added here, and it contains a pair of high energy electrons. What happens is these electrons are donated, gotten rid of, lost from NADH. The bonds rearrange in the structure and you end up with NAD+. And I'm going to draw it here just so we can see the distinction. I just realized I made a small error over here. It's not CH2, it's NH2. But what you can see here is, remember, all of each of these there's a hydrogen. I just didn't draw them, but let me go ahead and draw them just in case you're keeping score at home. And this one here used to have that extra hydrogen in it. It's now been lost. So this hydrogen right here, which comes off, and it's negatively charged, but the electrons are peeled off, and we end up with something that looks like this, where we created this proton, which we're going to use to establish that electrochemical gradient, and the electron itself here, which we're going to donate, that's going to generate the energy necessary for the movement of these protons. And then remember what's going to happen here, and I'm just going to draw it down here. Let me box it off though so we don't get it confused with what we have here. So remember, these electrons and protons are going to generate this electrochemical gradient that will help drive the ATP synthase. So as these electrons are being transferred, and we haven't talked about this yet, but we will go into a little bit more detail in a moment, they're transferred between these respiratory complexes. As they're moving through here, they're being transferred, these electrons are being transferred from one complex to the next. Every time they do that, hydrogen moves across to the other side. And then they will move back across. And as they move back across, these high energy electrons, they will finally interact with oxygen, which is the ultimate electron acceptor, to generate water. In that process, as I said on the last whiteboard, we make lots of ATP. What I'm going to spend a lot of time on in the next whiteboard are the actual enzyme complexes here. So let's talk about these respiratory enzyme complexes. And we're just going to call them these complexes, but remember that when we say complex, and this is very true in this case, these complexes contain many different proteins that work together to achieve the ultimate goal of moving these electrons. Who knew electrons could be so important? Okay, so let's go ahead and draw some membranes here. And what I'm going to do here is I'm only going to draw the inner membrane. I'm going to draw it like so here. Remember, our outer membrane, I guess I can draw it up here. The outer membrane doesn't play much of a role here, except that it creates this inner membrane space. We're going to spend most of our time talking about the inner membrane and what's happening there. At the inner membrane, we have a series of 3 respiratory enzyme complexes I want to talk about, and 2 other proteins I want to talk about. So here we have the first one, and this first one is called NADH dehydrogenase. And what NADH dehydrogenase does is it takes NADH and makes NAD+. In doing so, as you remember from the previous slides, and generates this proton, which needs to move across this membrane. But it's an energetically unfavorable reaction. It's going against the gradient. But it's able to go across this across this complex because it's taking advantage of this high energy electron that's also produced when NADH is converted to NAD+. And so we're going to draw this electron just like so. Now the reason the electron can move to this NADH dehydrogenase from NADH is because NADH dehydrogenase has a higher affinity for that electron shown here. Now the next complex I want to draw, I'll draw it right here. And this one is cytochrome C reductase. However, this electron can't just move over here. There are some mobile electron carriers that we have to talk about. And the first one we're going to talk about right here. And it's called ubiquinone. And it has a higher affinity for electrons than NADH dehydrogenase. So the electrons will move to it. And then, it being a mobile protein, it can now take those electrons and bring it to cytochrome C. So now cytochrome C has them. And I would suspect you could guess, why does cytochrome C get these electrons? It's because it has a higher affinity for electrons than ubiquinone. And during this process, we get another proton coming across the membrane. Now we have a third complex, and it's called cytochrome C oxidase. Now again, we can't just get these electrons from here to here. We need another one of these mobile carrier electron proteins. And so this one is called cytochrome C. These electrons move from cytochrome C reductase to cytochrome C, again because cytochrome C has a higher affinity for the electrons. It can then transfer these electrons from cytochrome C to cytochrome C oxidase. Again, a proton will move across this protein, again establishing a strong electrochemical gradient. This electron in this third complex, cytochrome C oxidase, will then be transferred to the last electron acceptor, and that is oxygen. So electrons are transferred to the oxygen and create water. So this here is ultimately, this very last step is the step where oxygen is needed for aerobic metabolism. A few things I want to point out here is that this movement is unidirectional. So we go in this direction, from this enzyme complex to this protein to this complex, protein complex. And the reason that the movement of electrons is unidirectional is strictly dependent on the fact that there's this change in affinity for electrons. NADH has the lowest affinity and oxygen has the highest. And we have this nice gradient going from NADH dehydrogenase to ubiquinone, cytochrome C reductase, to cytochrome C oxidase, then to oxygen. Each with an increasingly higher affinity for electrons, allowing the electrons to move to ultimately get to water, but to allow for the movement of protons to this inner membrane space. Okay, so what we've established here are a lot of hydrogens over here. If these hydrogens were just allowed to move back across the membrane all by themselves or through some protein similar to these, it would be released as heat. And there are some advantages to that. But for everyday normal functions, we don't want all this potential energy that is stored up here to just be lost to heat. Our cells and the cells of bacteria have evolved a mechanism to use these protons to generate ATP. So let's talk about that next. All right, so let's go ahead and draw this membrane again. And what we're going to do is going to talk about the importance of this protein complex called ATP synthase. It's perhaps one of the coolest enzymes that you'll ever learn about. As we'll see, it's this little miniature machine, this very ancient miniature machine that generates all this energy that we have used throughout the existence of life, aerobic life at least. Okay, so remember, this is our inner membrane here, and we can show our outer membrane up here just to keep everything legal. And remember, down here is our matrix. Inner membrane, outer membrane, and the inner membrane space. And remember, through the transfer of electrons that we had on the last whiteboard, we had a lot of protons over here, more than the cell knows what to do with. And at the closing of the last whiteboard, I said we could just move these all across here through some mechanism. And if we did that, all this potential energy would be lost as heat. And we'll come back to that later on, I think. I hope to, anyhow. But we want to talk about how the cell can use these protons to generate ATP. So we have this large protein complex shaped similar to how I'm drawing it here, where we have this main protein structure here, and then we have something we call the stalk. And attached to the stalk are a complex of proteins that we'll call the head. So we have the stalk here and the head here. What happens is that these protons will move across this ATP synthase. In doing so, they spin rapidly this stalk here. The head here remains stationary, but the stalk that's attached to this head will move really fast. As the stalk spins really fast amongst this complex of proteins here, these proteins will have a conformational change, which causes this unfavorable reaction, ADP plus PI, to move to ATP, the phosphorylation part of oxidative phosphorylation. Well, how does it do that? Well, what I didn't show you here is what these proteins here look like. So I'm going to try to draw this here in a way that will illustrate how this reaction occurs. So this would be a cross-section through these proteins. What happens is ADP plus PI are sitting together in these proteins. As the stalk spins them, they create a favorable interaction that forms the ATP molecule. And then as it keeps spinning here, this ATP is lost from the complex, released, and then new ADP, inorganic phosphate, come into this site here. And then every time, so let me draw that in here, ADP PI, once the ATP is left. So every time this spins, what happens is you get ADP PI being placed together in a favorable interaction because of this conformational change, which then forms ATP. As it keeps spinning, these conformation changes again and it kicks out ATP, allowing ADP plus inorganic phosphate to bind again. And it just keeps doing this cycle over and over again. So what's happening here is that we have this mechanical structural change. This mechanism changes the shape of these proteins. And the change of that protein generates a high-yielding chemical bond. We often think of this structure here, this ATP synthase, as the oldest and smallest rotary engine. Because of the rate of speed of how fast this stalk is spinning and the efficiency of this enzyme, it will produce more than 100 ATP per second. Now to just blow your mind a little bit more, because I know at this point you're enthralled in how awesome this enzyme is. Remember that this inner membrane is extensive throughout the cytoplasm. This mitochondrial network is extensive throughout the cytoplasm. And along this inner membrane, not only is it chock full of those oxidative phosphorylation enzymes, but it also has a lot of these ATP synthases. So each one of these is constantly making over 100 ATP per second. And all we have to do is give it a little glucose. It's not such a bad deal for us. Okay, now, if that didn't impress you enough about this enzyme, I'm going to tell you something else it does. And that is that the protons don't always move in this direction. The protons can move in this direction. And when it moves in this direction, it converts ATP into ADP plus inorganic phosphate. So depending on which direction the protons are moving will alter the direction of this complex. They go this way, it spins one direction and hydrolyzes ATP. It spins the other direction because the protons are moving this way. It generates ATP. So this is a very fascinating enzyme in the sense that it can both create and hydrolyze ATP. Okay, I do want to talk a little bit about the overall yield that we're going to get from glucose, but I just remembered as I was thinking about the yield that I forgot to tell you something. And it's pretty important. Remember when we were talking about oxidative phosphorylation and these complexes here, where we had the NADH dehydrogenase here. So I'm going to write down here NADH dehydrogenase, so DH, and then cytochrome C reductase, and then cytochrome C oxidase. We also had the quinolones, and we also had the cytochrome C. And I mentioned to you that NADH enters here to generate NAD plus, to move the protons across here, and then to transfer the electrons down the way. Well, I didn't mention what FADH2, because remember from the citric acid cycle, we make one molecule of FADH2. FADH2 doesn't donate its electrons here. It donates its electrons to ubiquinone, which I miswrote there, ubiquinone. So it doesn't enter into this electron transport chain at the same location. And because of that, it doesn't generate as much hydrogen protons across the membranes. It's only 2/3 as much, or protons going across the membrane. So that makes a difference when we start calculating how much ATP is going to be made from one molecule of glucose. So what I want you to do and think about on your own is, what is the yield of ATP from one glucose molecule? What you're going to want to do as you're calculating this is to think about glycolysis, the citric acid cycle, and oxidative phosphorylation. How much ATP is made from each of these per molecule of glucose, how much NADH is made, and how much FADH2 is made from one molecule of glucose. We know about, there's some debate about this, but we get about 3 ATP from each NADH and about 2 ATP from each FADH2. So what you want to do is figure out how many NADH are made, how many FADH2s are made, multiply them by these numbers appropriately, and then how many direct ATPs are made in these 3 cycles. And then that'll tell you your total yield. Okay, on that, I'm going to end this podcast on oxidative phosphorylation. If you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.